All right, welcome to the lecture on fatty acid catabolism. And because this lecture is going to be focused on the catabolism of fatty acids, what we're really discussing here is how we are mobilizing fatty acids stored in adipocytes, overwhelmingly in the form of triacylglycerols, and how the energy that's stored becomes mobilized, distributed throughout the organism from a eukaryotic perspective, and then how those free fatty acids are then broken down sequentially to release the energy available in them. And the reason this is so important biologically is because fatty acids are the most efficient mechanism by which we store energy. Yes, carbohydrates have energy at about approximately 4 kilocalories per gram, and proteins similarly have about 4 kilocalories per gram, but fatty acids have about 9 kilocalories per gram, and that's because fatty acids have a more reduced structure to the carbon chain. So fatty acids, 3, 4, 5, 6, 7, 8, 9, 10, have a structure something like this. Whereas a carbohydrate, let's say glucose, has a structure like this. And the big difference between these, from an oxidative standpoint, is that a vast majority of the fatty acid chain, of this C18 chain, is not oxidized. Obviously, we have the carboxylate on the C1 carbon, but carbons 2 through 18 in this example just have hydrogens. There's no oxygens on them. So if all metabolism tends towards the same product of, son of a gun, the same product of CO2, which is the most oxidized form of carbon, there is more room to abstract energy from these reduced carbons on the way to CO2 than there are from this one, because the carbohydrate carbons are already more oxidized. So there's less delta between their initial state and the final state terminally achieved in oxidative metabolism. So that's why we get about 9 kilocalories per gram from a lipid, and we get about 4 from the carbohydrate or the protein. That said, let me get rid of this. I love how the tablet tries to remind me that I can erase faster, and by virtue of reminding me, slows the speed with which I can erase. So, back to the core topic here, fatty acid catabolism. Most of the fat storage in our cells are stored in adipocytes, or fat cells, and in the form specifically of triacylglycerols. And let's recall that a triacylglycerol has 3 different fatty acid components, each linked to a glycerol backbone. And what that looks like is we have, drawing bonds too early here. And then let's see here, I'm not counting here and I'm not making nice corners. So I just have to use your imagination that those are fatty acid tails. And you can see that here we have the ability to cleave free fatty acids from our glycerol backbone. So this is how we're storing fatty acids as triacylglycerols in adipocytes. Erase this. But how is this energy mobilized? So what mobilizes this energy biologically in the form of stored energy is that hormones are going to trigger the release of fatty acids from adipose tissue. And the way this works is that fatty acids are mobilized from adipocytes in response to hormone messengers, which include adrenaline, glucagon, and adrenocorticotropic hormone. So we have adrenaline, glucagon, my goodness. I know everyone's laughing at my lack of tablet skills here, excuse me. And adrenocorticotropic. Hormone. Right. And what happens is these signaling molecules bind receptors on the plasma membrane of the adipose cells and leads to the activation of adenylyl cyclase, which forms cyclic AMP, or CAMP, from ATP, which is one of our classic second messengers that we discussed last semester. So in adipocyte cells, cyclic AMP activates protein kinase A, which then phosphorylates and activates a triacylglycerol lipase, which is a hormone-sensitive lipase that hydrolyzes a fatty acid from the triacylglycerol. So what this looks like, let's go ahead and make a little bit of space here, is let's say we have the outer membrane of this fat cell, and we have some receptor on the cell, and one of these hormones that we discussed comes in and touches the receptor. And it could be the adrenaline, the glucagon, the adrenocorticotropic hormone. And what's going to happen here is this receptor is coupled to adenylyl cyclase. My goodness. And what this does, once activated, is it converts ATP to cyclic AMP. As well as releases pyrophosphate. What the cyclic AMP then does is it activates protein kinase A. Via phosphorylation. And what the pKa then does is it phosphorylates as a kinase, activating a triacylglycerol lipase, which hydrolyzes the fatty acid. So what this looks like is triacylglycerol lipase, and we'll just call this inactive. And then through phosphorylation, I'll just write active here. And what this is going to do then is act on triacylglycerols, liberating fatty acids. So let's say we have an acyl backbone and we have our triacylglycerol with 3 acyl chains or 3 fatty acids. And what will happen is the triacylglycerol will then lose one of its fatty acids and end up as a diacylglycerol. Now, subsequent actions include an action of a diacylglycerol lipase and then a monoglycerol lipase. Each of these are going to liberate another fatty acid, eventually just giving us a glycerol backbone that could be reutilized later. And then following the liberation of these free fatty acids, we need some way to mobilize them. And at some point, you've probably wondered, how do we get free fatty acids to mobilize in the bloodstream? Because fats typically don't mix with water, and they typically not only don't mix, but formally separate from aqueous solutions. And this is the case as well. In this case, though, of course, we have the polar head group, so it has the capacity to form micelles as well as vesicles. But that's another issue entirely. So how do we mobilize free fatty acids? Well, the answer happens to be we have a very important protein, serum albumin. Which happens to be the most abundant protein in blood serum. And what serum albumin primarily does is transports free fatty acids to sites of utilization. So it's the conduit which binds these free fatty acids and mobilizes them throughout the system. So it helps deliver free fatty acids for energy consumption as well, because it has to get from the adipocyte to the thing that needs it. And it also helps mobilize digested free fatty acids for storage at the adipocyte. So incredibly important protein. So let's make a little bit of space here, so we can talk about what happens once we've liberated fatty acids. Well, the real question is how do we take free fatty acids and convert them to energy? How are free fatty acids catabolized? Or, in essence, how is it broken down? And this is going to come down to a process called beta oxidation. So beta oxidation, in terms of elucidating it, goes back to the early 1900s, where it was discovered that fatty acids have to be degraded by oxidation at the beta carbon. By the beta carbon, what we're talking about is if we have a long chain C18 chain, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18 carbons, the alpha carbon is the first carbon next to the carboxylate. The beta carbon is the carbon second downstream from the carboxylate carbon. And the cleavage happens at this point, yielding 2 carbon units. And it was originally thought that this was just liberated as acetyl units or acetyl units. However, it was later found out that these are being released as acetyl-CoA. And because this cleavage happens at the beta carbon, the term for this process was called ultimately beta oxidation. Now, in mammalian cells, which we're going to be primarily focused on, beta oxidation takes primarily place in mitochondria, but similar pathways also take place in peroxisomes. We're going to focus primarily on mitochondrial processes for the sake of these lectures, though. And it's the mitochondrial beta oxidation which primarily provides energy to the organism, whereas peroxisomal beta oxidation is responsible for shortening long-chain fatty acids that are poor substrates for mitochondrial beta oxidation. So they assist in the process, but most of the core of the catabolic pathways, which we're going to focus on, are going to be the mitochondrial processes, because the peroxisomal processes really are just about shortening chains so that they can feed into mitochondrial beta oxidation. Now, a couple other qualifiers before we really jump into this. Coenzyme A is what activates fatty acids for oxidation. And you probably see this CoA and you had a suspicion that it might act as an activating component, and that's the case. So that should be an A, not a triangle. See, every time I do that. Yep, see, there we go. I'm going to have to hang at this. I have to have my hand hovering over the tablet, I'm not sure. There we go. Activates fatty acids for beta oxidation. So the process of beta oxidation really begins with the formation of a thiol ester bond between the fatty acid and the thiol group from coenzyme A. And this reaction is catalyzed by acyl-CoA synthetase, which has a couple other names, but I'm going to refer to it primarily as acyl-CoA synthetase. You may see acyl-CoA ligase or fatty acid thiokinase as well. And this condensation with coenzyme A activates the fatty acid for reaction in beta oxidation pathway. Now, before we get into that, because we're going to take a look at it, for long-chain fatty acids, this reaction normally occurs in the outer mitochondrial membrane in higher eukaryotes before entry of the fatty acid into the mitochondrion, but it may also occur at the surface of the ER, endoplasmic reticulum. Now, the short or medium-length fatty acids undergo this activating reaction in the mitochondria specifically. So you have two different locations that this takes place. But in all cases, the reaction is accompanied by the hydrolysis of ATP to form AMP in pyrophosphate. So let's take a look at these reactions. Firstly, let's take a look at the activation step. So the activation step looks like this. I'm going to have our fatty acid, and I'm not going to count them. This is going to be an approximate. Tail is probably going to be overly short, and we've got our carboxylate, and this is going to react with coenzyme A, which includes a complete sulfohydryl group and ATP. And what happens, and note this is going to be listed as reversible because of the thermodynamics. We get our activation step as well as the production of AMP plus pyrophosphate. Now, what's really important to note thermodynamically is that this first step that's noted by equilibrium has a net delta G of about negative 0.8 kilojoules per mole. So yes, it's technically slightly favorable. However, it's not really that far off of zero or an equilibrium process. So even though it's slightly favoring products, it's genuinely a reversible process where it's not at equilibrium, but it's close. Now, what's important though here is that the pyrophosphate, this paraphosphates, undergoes a second spontaneous hydrolysis in the presence of water, which biological systems tend to have plenty of, where it's separated into a separate pair of phosphates. And this has a delta G of negative 33.6 kilojoules per mole. So it's highly favorable. And because this is a natural outcome coupled to the first, this is what really motivates the first reaction towards products. And please recall that this is being facilitated by acyl-CoA synthetase. Okay, so now that we've got these activated fatty acids, what happens next? Well, the next really important step is that carnitine is going to carry our fatty acyl groups across the inner mitochondrial membrane. What's that look like? Well, So that is written, to restate it, carnitine is responsible for carrying fatty acyl groups across the inner mitochondrial membrane. Why is this so important? Well, all of the other enzymes of beta oxidation pathway are located in the mitochondrial matrix. Short-chain fatty acids, which we've already mentioned, are transported into the matrix as free fatty acids and form the acyl-CoA derivatives there. However, long-chain fatty acyl-CoA derivatives can't be transported into the matrix directly. These long-chain derivatives must first be converted to acylcarnitine derivatives, which we're going to take a look at in just a second. And it's carnitine acyltransferase 1 associated with the outer mitochondrial membrane, which catalyzes the formation of orthoacylcarnitine, which is then transported across the inner membrane by translocase enzyme. And it's at this point that the acylcarnitine is passed through yet another carry, carnitine acyltransferase 2, on the matrix side of the inner membrane, which transfers the fatty acyl group back to CoA to reform fatty acyl-CoA, leaving free carnitine, which can then return, so it can retransport yet another fatty acid. So what's this look like? Let's take a look at it. Let's make a little bit of space. So firstly, we have to draw our outer mitochondrial membrane. And we'll put our carnitine acyltransferase 1 here. Remember, this is our outer membrane, outer mitochondrial membrane. And then we have our intermembrane space and our inner mitochondrial membrane. And we'll put this over here. And this is going to have our acylcarnitine translocase. And in addition to that, we'll have our acyltransferase 2. So carnitine acyltransferase 1 is on the outer membrane, and the 2 is on the inner membrane. And what this looks like is that carnitine will interact with the CoA-activated fatty acid at the carnitine acyltransferase 1 in the cytosol. So let me draw that. I'm actually just going to erase this and make a little bit more room because it's a little tight and there's no point in making this harder than it needs to be. So we've got, let's see, our carnitine. And then we've got our fatty acid, which has been activated. And what's going to happen is the carnitine is going to kick off the coenzyme A sulfohydryl. So this comes in, our coenzyme A kicks off, and what we end up with transferred through to the intermembranous space between the outer and inner membranes is our acylcarnitine derivative, where we have our free fatty acid, which has the carbon, the oxygen, and then it's bound where this hydroxyl group was on the carnitine. We've got our positive nitrogen with our 3 methyls. And this is our acylcarnitine. And then the acylcarnitine is shuttled through to the mitochondrial matrix via translocase, where it interacts with carnitine acyltransferase 2, which kicks off the carnitine and adds the coenzyme back. So we just reverse those first steps. So this, removes the carnitine and adds our coenzyme back, reforming the activated long chain acyl-CoA. So now that everything is where it's supposed to be, we've got fatty acids liberated from adipose tissues by hormonal signaling, breaking down our triacylglycerols, the free fatty acids, binding albumin, making it to the cells that need energy. Those cells take that up. It gets activated. Free fatty acids are activated by interacting with our coenzyme A, being facilitated by acyl-CoA synthetase. Once we have it activated, it's transported into the mitochondria through these series of events where the long-chain fatty acids are modified via carnitine. And now everything we have is back in the matrix where it needs to be, and everything is reactivated. So now we're at beta oxidation. So let's go ahead and make a little bit of room. So beta oxidation really involves a repetitious sequence of 4 reactions. And our 4 reactions include an oxidation step, a hydration step, another oxidation step, and a cleavage step. And then the whole process repeats again. So I'm going to go into this in a lot more detail in a second. But for saturated fatty acids, the process of beta oxidation, like I said, involves this recurring cycle of 4 steps. And the overall strategy in the first 3 steps is to create a carbonyl group on the beta carbon by oxidizing the bond between the alpha and beta carbons to form an olefin with subsequent hydration and oxidation. So, in essence, the cycle is directly analogous to the sequence of reactions converting succinate to oxaloacetate in the TCA cycle. So, biologically, we're using the exact same chemical strategy to make this happen, as we've already seen, going from succinate to oxaloacetate. So then the fourth reaction, because that's the first 3, the fourth reaction of the cycle cleaves the beta keto ester in reverse Claisen condensation, which gives us the yield of our acetate unit and leaves a fatty acid chain that is now just 2 carbons shorter than it began. And then that re-enters the initial point of the cycle and repeats and repeats and repeats until we've abstracted all of the 2 carbon units that we can from that fatty acid chain. So let's go ahead and illustrate beta oxidation, which I think will give everyone the best picture of what's going on. So let's start with some generic fatty acyl-CoA, where we have some fatty acid chain R. We have our beta carbon, our alpha carbon, our carbonyl carbon, and our coenzyme A. And we should probably put our hydrogens in on here because it's going to matter in a second. Okay, so in the first step, it's going to be our oxidation step, our first oxidation step. And we're going to get a unit of FADH2 out of this. So let's see, I need to give myself enough room. And the enzyme in this step 1, which is our oxidation, is going to be facilitated by acyl-CoA dehydrogenase. and we're going to yield an equivalent of FADH2 from this oxidation step. And what we're going to do is we're going to add electrons between the alpha and beta carbon, forming a double bond. And it's going to look like this. Oh, starting out, we should probably label it, fatty acyl-CoA. Notice that we still have our beta carbon and alpha carbon, but we've removed one proton from each of them. That's why we have FADH2 now. And we've reduced our FAD to FADH2, and we've oxidized the bond between our alpha and beta carbons, forming a double bond. We now have, let me actually finish drawing this before I start from my mouth, trans-anoyl-CoA. And it's trans because the protons are trans to one another on the newly formed double bond. Now, from this first step being complete, we undergo a hydration step, very similarly to that referenced reaction in the TCA cycle. And this is going to use the enzyme anoyl-CoA hydratase. So, very nice descriptive name. In addition to the hydratase, we need water if we're going to hydrate a double bond. And step 2, if you can recall, is a hydration. So everything about this is screaming, we're putting the water across a double bond. And what that looks like, in this particular case, is adding the hydroxyl to the beta carbon. This is now beta hydroxyacyl-CoA. And this absolutely makes sense because we've put a hydroxy group on the beta carbon, so beta hydroxyacyl-CoA makes sense. From here, the third step is our second oxidation, and what we're going to be doing is oxidizing the newly added hydroxyl group to a ketone. And this is going to be utilizing the enzyme hydroxyacyl-CoA dehydrogenase. And I'm running out of room, but we're going to go, in this particular case, because we have a single hydrogen to get rid of. That's not quite true. We have 2 hydrogens to get rid of. We have one here and one here. And we're going to, in this case, utilize coupling this reaction to NAD plus, going to NADH plus a proton. And that accounts for our 2 protons that we are losing from our beta hydroxyacyl-CoA, and we're going to yield a beta ketoacyl-CoA. So everything's the same, except that now the beta carbon has a ketone on it. Let me write the name down, beta keto. There we go. And that's 3 steps down. What we've done is strategically at this point taking our fatty acyl-CoA, and we have abstracted energy in the form of FADH2 and NADH going from a saturated bond between the alpha and beta carbon to oxidizing it to a double bond, adding water across that double bond, and then doing another oxidative step to that hydroxyl group, forming a ketone. And now that we have a beta ketone, notice that we've put a ketone on either side of the alpha carbon. What this does, let me see if I can get rid of this now, is pulls electrons towards both of these carbonyls. And this makes the carbon between the pair of carbonyls electron poor, which makes cleaving this bond between the alpha and beta carbon substantially easier energetically than it would be otherwise. So this is the entire strategy here, is to make a pair of carbonyls on either side of the alpha carbon so that we can cleave the bond between the alpha and beta carbon in an energetically efficient fashion. We've really primed it to lose this acyl group. This acetyl group. So from here, we have our 4th step, cleavage. Which makes sense because that's exactly what we've primed. And at this point, we come in with an equivalent of coenzyme A using the enzyme thiolase, and it's going to basically do an attack on this carbonyl at the beta carbon, which is going to add a new acyl-CoA to the beta carbon, kicking off the acetyl group, the acetyl-CoA, as a leaving group. So we've got our thiolase enzyme, we have our coenzyme A coming in, and what's going to happen is we're going to say, let me pull that back up again. Let me see, maybe a different color this time. We're going to come in with that coenzyme A. Let me see if I can make this a little bit, get rid of these reds. Come in here with a nucleophilic attack the carbonyl. Electrons are going to come up, they're going to come down, they're going to kick this off as a leaving group. We're going to have added a new coenzyme A to this carbonyl, forming a new acyl-CoA, that's 2 carbons shorter, and we're going to be kicking off this entire group here. So, getting black back. Coming off is our acetyl-CoA, where we have a, let's see here, CH3, COS, COA, and that's our leaving group, and that's our 2 carbon unit that left, and this was originally our alpha carbon, and now what we're left with is a fatty acyl-CoA, which has been shortened by 2 carbons. So I can redraw this product. And just, even though this isn't actually out the beta, it was originally the beta carbon, but now it's the new carbonyl carbon, and the one next to it is the alpha, and the one next to that is the beta. So this is minus 2 carbons. What happens now is we have a new starting reagent. So we had a fatty acyl-CoA to begin with, and we still have a fatty acyl-CoA, and this now re-enters the first step of oxidation with acyl-CoA dehydrogenase. We form a new double bond, we put a water across it, we oxidize that hydroxyl to a ketone, and then we cleave, and then we've lost another unit of acetyl-CoA. Now that we've lost that acetyl-CoA, we've down 4 carbons. This chain re-enters again. and again. And every single cycle, we are getting another equivalent 2-carbon units lost, acetyl-CoA, and for each cycle, we're also getting an FADH2 and an NADH out of this. And that's really beta oxidation in a nutshell. So let's talk briefly about the energetic consequences of beta oxidation. So what we've seen is that the repetition of these cycles of beta oxidation yields a whole bunch of different acetate units. In essence, this sequence of 4 reactions has yielded a fatty acid as a coenzyme A ester that's been shortened by 2 carbons and 1 molecule of acetyl-CoA, and this happens over and over and over again. And for each of these cycles, let's say we go all the way through beta oxidation for, say, palmitic acid. This would give us, which has 16 carbons, this would yield 8 molecules of acetyl-CoA as well as 7 molecules of FADH2 and 7 molecules of NADH. So the acetyl-CoA can further be metabolized in the TCA cycle, which we've already seen. And alternatively, acetyl-CoA can also be used as a substrate in amino acid biosynthesis. So we've yielded FADH2, 7 per palmitic acid. We've yielded NADH, 7 per palmitic acid. We have 8 equivalents of acetyl-CoA, which each feed into the TCA cycle. So if each of those were fed into the TCA cycle versus other synthetic routes in the mitochondria, it can eventually generate about 10 high-energy phosphate compounds, which would include ATP, as well as FADH2 and NADH. All said and done, complete beta oxidation of a molecule of palmitoyl-CoA, simple fatty acid, in a mitochondria will yield about 108 molecules of ATP. As an energetic consequence of a single free fatty acid, making its way all the way through. Now, to be clear, there are a couple important branches off of this. Note that there are some fatty acids that have odd numbers of carbons. And so there are some special pathways for dealing with odd number carbons, utilizing propionyl-CoA carboxylase and some other enzymes. We're not going to go into those pathways. I want you to focus on what we've looked at here, and also recognize that this has been entirely focused on saturated fatty acids. Of course, we have unsaturated fatty acids which have double bonds in them, and there are some specialized pathways which include reductase enzymes which allow us to deal with those double bonds prior to them entering the beta oxidation pathway. So know that there are some additional tools in the toolbox biologically for dealing with unsaturated fatty acids as well as odd numbered carbon chain fatty acids. But in the context of what we're doing for this class, let's stay focused on the content of this lecture, and what we'll do next is be picking up a discussion of the other side of fatty acid synthesis. Excuse me, not fatty acid synthesis, lipid biosynthesis. So we just talked about the catabolic processes associated with lipid metabolism. Next, we're going to focus on the anabolic processes of lipid metabolism. So I'm going to get this one loaded up, and we'll be discussing lipid biosynthesis next.